Hello guys, welcome to session 3. In last session we are discussing about the pollen grains and discuss about the gynoecium and its parts. So in this session we are going to focusing on the megasporogenesis or ovule. So especially this ovule we are calling as a anatropous ovule. Okay, so ovule, it is considered to be an integumented. Now what is the meaning of integument? Means nothing but it is a protective layers. So here the ovules are covered by the protective layer, right? So it is a integumented megasporogenesis. Each ovule, it is a earlier or first develops as a small projection. So it is develops as a small projection on the placenta. So it is show a small projection on the placenta. So this placenta, okay, so this placenta, it is connected to the ovule by the help of stalk, and that stalk you are calling as a funicle. That stalk you are calling as funicle. So this ovule earlier, it consists of a mass of parenchymatous cells. Before complete development of ovule, so initially it consists of a mass of parenchymatous cells called nucellus. It's called nucellus. So later the growth and development of ovule proceeds. As the growth and development of ovule proceeds, so this nucellus is rise on the stalk-like structure. Yes, the ovule, as per development of ovule proceeds, so nucellus is rise on the stalk-like structure. Okay, so that is funiculus, or we are calling as funicle. So here funicle is nothing but it is a stalk where the ovule will attach to the placenta. So this stalk structure we are calling as a funicle. So this ovule, it is covered by two protective layers. As we already discussed that it is integumented. So integument nothing but it is having protective layer, right? So it is surrounded by one or two protective layer called integument. So which grow up from the base of the nucellus. So from which part these are arising means these are arise from the base of nucellus called chalaja. So integuments ಎಲ್ಲಿ start ಆಗ್ತವೆ ಅಥವಾ ಎಲ್ಲಿಂದ ರೆಡಿ ಆಗ್ತವೆ ಅಂದ್ರೆ it is from the part that is a chalaja. Okay, so these integuments leave a small opening. So we know that integuments nothing but it is a protective layer which will initially it is a start from the base of the nucellus, that is what we are calling as chalaja, and it is not completely covered. It is not completely covered to the entire ovule, but it leaves the somewhere small gap, and that opening you are calling as a micropyle. That opening you are calling as a micropyle. So now what is the significance? So now what is the use of this micropyle? Means this opening is as a passage for entry of pollen tube. Yes, it's provide passage for what? Pollen tube into the ovule. Okay, the point at which body of the ovule is attached to the funicle. The point at which the whole ovule body is attached to the funicle is called hilum. That part we are calling as a hilum. We can observe in diagrams. Okay, I'm going to repeat again. So we're discussing about megasporogenesis. So nothing but it is a ovule. Okay, so ovules are we are calling as integumented megasporogenesis because it consists of integuments. So we can observe in diagrams, right? So there are two integuments are here. One is outer integuments and one is inner integuments. Okay, so earlier so it consists of what? A mass of parenchymatous cells, right? It is developed from what? They are developed from the mass of parenchymatous cells, and those cells we are calling as a nucellus. As the growth and development of the ovule proceeds, the nucellus is rise on the stalk. Yes, and it produces a structure, and that structure we are calling as a funicle. So later, so as on development goes on or proceeds, so these ovules are covered by the two protective layer, okay, which grow up from the these two protective layers which grow up from the base of nucellus, and that part we are calling as a chalaza. Yes, we can observe in diagrams, okay. So these integuments is not covered entirely. Somewhere they are having the small gaps or opening, and that opening you are calling as a micropyle. Now what is use of this micropyle? Means it is helps in the, yes, it is helps in the, or it make a passage entry of pollen tube into the ovule. So the point at which ovule is attached to the funicle, and that part we are calling as a hilum. So below the diagrams we have mentioned the note, right? So a typical embryo sac contain 8 nuclei. Typical embryo sac contain 8 nuclei but 7 cells. So 3 cells in agapetus, 3 cells in antipodal cells, and 2 cells in polar nuclei. Okay, so we'll discuss about this. So when the inside the embryo sac, it is undergoing the successive mitosis divisions, and totally it will form the 8 cell, and these 8 cell they are get distributed, and 3 cells are get accumulated in the micropellar regions, and 3 cells are get settled in the chalazal end. So remaining 2 cells are present in the center of the embryo sac. So these cells later it will fuses and then become single cell. So because of this we are calling as it is a 8 nuclei. When the divisions occur, it is a 8 nuclei. But later what happens? They are become, it is a 7 cells. In this slide, we are going to discussing about the megasporogenesis, right? So megasporogenesis means it is a process of formation of megaspores from megaspore mother cells is called megasporogenesis. So ovules generally differentiate single megaspores. Ovules generally differentiate a single megaspore mother cells, that is we are calling as MMC, in the micropylar region, in the micropylar regions of the nucellus. So we know that these ovules consist of a number of parenchymatous cells, that is we are calling as a nucellus. Okay, so it is a large cells containing dense cytoplasm. It is a large cell containing dense cytoplasm and a prominent nucleus. It consists of, yes, prominent nucleus, which is a large number of dense cytoplasm. So this megaspore mother cells undergo meiotic divisions, and it will result in the production of 4 megaspores. So megaspore mother cells undergo meiotic division and produces how many spores? 4 megaspores. Okay. Out of 4 megaspores, 3 are get degenerate, and only one it will develop into embryo sac. And this type of development we are calling as a monospermic. Yes, we know that there are 4 megaspores are produced. Out of 4, 3 are degenerate, only one will be get developed into embryo sac, and this method or this thing you are calling as a monospermic. This embryo sac, it contains of nucleus, and this nucleus undergo 3 successive mitotic divisions. Okay, the nucleus undergo 3 successive mitotic division, and it will form the haploid nuclei. It will form haploid nuclei. So the megaspore enlarge. The megaspore enlarge into oval shaped structure called embryo sac. Okay, so this embryo sac This embryo sac consists of 8 nuclei. They arrange themselves into 3 groups. So one is it is a antipodal cells, one polar nuclei and egg apparatus. Okay. In last slide we discussed about the megasporogenesis. In this slide we will discuss by the diagrammatically that megasporogenesis process. So we can observe in first diagrams we know that initially ovule will consist of what? Parenchymatous cells. Okay. So these parenchymatous cells in first A diagram you can observe that the parenchymatous cells which is near to the micropylar region it become dense and large size compared to other parenchymatous cells. It will become large and it consists of prominent nucleus and that is we are calling as a megaspore mother cells. So where it is found near to the micropylar regions, out of many nucellus only one cell it is become large with prominent nucleus and that is we are calling as a megaspore mother cell. These megaspore mother cells undergo meiotic division and produces 4 megaspore cells. It produces 4 megaspores. Okay. Out of 4 megaspores, 3 are get degenerate. 3 are get degenerate. So there is only one megaspore cell, and this process we are calling as a, or this thing we are calling as a monospermic. That is we are calling as monospermic. So later this monospermic cell which undergo successive mitotic divisions. Later it undergoes successive mitotic divisions. So we can observe in the below diagrams. So first one showing the first mitotic division, second one showing the 4 cells means it is a second mitotic divisions. Third diagram showing the 8 cells, so it is a third mitotic division. So like this that megaspore mother cells undergo mitotic divisions and produces 8 nuclei, and these 8 nuclei they arrange themselves. Yes, they are arranged themselves and it will form the parts like chelazal end or antipodal cells. Yes, in micropylar regions it will form the egg apparatus and in central portion we can find the polar nuclei. So like this there is a formation of, yes, there is a formation of embryo sac. Okay, and this whole process we are calling as a megasporogenesis. So in this slide as we mentioned that is a homework. Okay, so homework is the first question, draw a neat label diagram of young anther. Second question is explain the steps involved in the sporogenesis. Third question is draw a pollen grain structure. Fourth question, write the effect of pollen grains. Fifth question, draw the structure of anatropous ovule. Next question is, write the steps involved in the megasporogenesis. Write the step involved in megasporogenesis and explain the briefly types of ovules. Explain briefly types of ovules. So this question will be comes for NEET question or competitive exam question. Okay. So last question, differentiate between the megasporogenesis and microsporogenesis.